then link them through an enzymatic ligation to other more interesting protein structures, again, with the goal of understanding how these receptors, answering questions or asking the questions about how more than one receptor may dimerize or interact on the surface, questions that are not as easily answered without the presence of the membrane. So, and the last few examples are from another researcher at IBBR, Ella Mikulescu, who's actually here in the audience, so she's right there in the second row, and she provided me with a few examples of again, looking at reflectometry to understand, to localize and understand peptide interactions with the membrane. So the first example is there's a number of toxins which interact with the potassium and other ion channels, and basically there's a large interest in understanding how these toxins actually interact and inhibit these ion channels. So as you've already seen in a number of slides, this is an experiment that can be easily amenable to the bilayer. You basically can generate bilayers, add the toxin, and then sort of do reflectometry measurements to sort of ask where does the density come in and where are these peptides located. And so again, this was able to demonstrate that the binding to the membrane and the localization of this toxin was at the surface of the membrane. And so, again, you can further show that the position and orientation of that membrane at the surface through neutron diffraction measurements can be ascertained. So again, the depth to which that toxin penetrates the membrane and its orientation relative to the membrane could be ascertained from both the diffraction and reflectometry. And again,